Welcome everyone to this week's edition of the Extracellular Vesicle Club. This is an event of the International Society for Extracellular Vesicles, and my name is Kenneth Witwer. I will be your host for today. And before we get started, just a reminder to everybody that during the presentation, you will be on mute, but you can ask your questions and also put your comments into the chat box. And then at the end of the presentation, we will allow unmuting so that you can interact with our guest today. And so our guest, I'd like to thank Jeff Sundstrom for volunteering to go this week. We had a gap here in the schedule, and Jeff stepped up and kindly offered to share some of his exciting research on the retina. So EVs and retinal diseases. Jeff is at Penn State Hershey, and he was just telling me about how the Penn State Hershey campus has some of the really the major resources that you need to do EV research. So Jeff, thanks again for joining us today, and we're looking forward to hearing about your work.
Okay, well, thanks so much for having me today. Very happy to share my work. We're a very open and collaborative group. I'm going to start today by just kind of sharing what our experience is with our laboratory. This is what we ask folks to do with our EV group. And so we've worked, when we first started getting into exosomes and EVs, we worked really hard on developing really robust protocols for EV isolation. I'm sure that the group here knows that that's absolutely critical to any generating any quality data. We're fortunate to have the NanoSite instrument in our lab. We have a cryo-EM, a TEM facility, and also atomic force microscopy suite. I've been working on doing proteomic analysis of Vitreous and as well as EVs. And that's one thing I'm certainly open to collaborative work with. So we can do quantitative proteomic profiling using a 10 or 16 plex. And we've developed algorithms to integrate that large data set across those multiple plexes into a single data set. I'll touch on that briefly. And then we've also done RNA-seq on EVs. So just a brief outline. I'm sure this is a mixed audience. There's not too many people that do EVs in relationship to the eye. So I'm going to talk about a little bit about the eye, and then I'll move over to some more basic science research about the EVs and how they impact RPE dysfunction. So just some basic eye anatomy to begin. Obviously light comes through the front of the eye, and the retina is like the film in the camera. The fluid that I became interested in as a graduate student was the vitreous fluid. I like to think of it sort of as the appendix of the eye. Retina surgeons remove it all the time for various reasons, like retinal detachment, non-clearing vitreous hemorrhage, and simply replace it with balanced saline solution. Generally thought to be an inconsequential fluid of the eye. On the right, you can see a picture of the retina. Here's the optic nerve, this is the macula, and here's the fovea. That's what I can see when I look at your eye in the clinical setting. Just to make sure everyone's up to speed with how retina, most retina patients are treated today, especially patients with age-related macular degeneration and diabetic retinopathy. That makes up the bulk of our patient volume. Anti-VEGF treatment, which is vascular endothelial growth factor, has been the gold standard since 2006. It really revolutionized how we treat patients with retinal disease, with AMD and DME. It involves an injection of an antibody into the vitreous fluid. And today, about 80% of patients in revenue are more what we call medical retina, as opposed to surgical retina. I'm just going to show you a brief case to sort of show you how we put the EVs into context here. So this is a patient with age-related macular degeneration of the right eye, some yellow spots that we call drusen, and some pigmentary changes that are a little tough to see. Those are hallmark lesions of dry age-related macular degeneration for which there is no treatment. When I saw this patient the day before, they had a subacute vision loss in the left eye and converted to hand motion vision. So when we inject these patients with the anti-VEGF antibody, you can see in the top photograph where we're able to take a optical section in the clinic using a device called OCT. There's a group of blood vessels here that are leaky, creating a massive amount of distortion and vision loss in the macular and foveal region. With one injection of anti-VEGF treatment, the patient was able to see 20/60. So definitely changed how we treat patients with AMD. Similar results are seen with diabetes. Unfortunately, across many large clinical trials, about only half of these patients respond really well with improved vision. And sort of the field is really wanted to know, how do we identify responders and non-responders? And I'm certain that there's many other, given that monoclonal antibodies are much easier to make, there are more antibodies being made all the time and being tested for the treatment of retinal disease. Most recently, a bivalent antibody has become available that targets both VEGF and Ang2. So as a graduate student, I was very frustrated that the animal models of diabetic retinopathy, which is what I worked on for my PhD, they don't really recapitulate the human disease state very well. And I had done some proteomics work, and this concept of a proximal biofluid or liquid biopsy was just starting to evolve. So it's not like cancer where we get cancer tissue from the patient and we can bring that back to the lab and look at the pathways involved in disease pathogenesis directly. And so I asked some of my colleagues to start saving the vitreous fluid that they were removing in the operating room. Here's a picture of the eye with vitreous, the way most of us think of it, as a solid organ within the eye. It's very gelatinous, primarily made of collagen and hyaluronic acid, and generally thought to be protein poor. So we started just doing some unbiased proteomics research. Admittedly, this was a very simplistic study, but I wanted to test the hypothesis that if you did unbiased proteomics from patients with proliferative diabetic retinopathy, go through your workflow and compare that to control. At this point in time, mass spec-based proteomics was very expensive, so we really we pulled these. So I know it's not a large study. We were able to find about 1,000 proteins in both control and proliferative diabetic retinopathy. And then when we ran the pathway analysis through IPA, which it supported and confirmed our hypothesis that just by looking at vitreous, you can see that proliferation of cells was enriched. And this Z-score here means that the pathway is activated if it's positive and inhibited if it's negative. So these patients have proliferation of endothelial cells, as you can see here. And my colleagues Tom Gardner and Alistair Barber had shown in the late 90s and early 2000s that there's also a neurodegenerative component with diabetic retinopathy. And you can see that there's downregulation of pathways that support neurogenesis and the process of synaptic development. So we were really impressed that just by this one simple experiment, we were able to show vitreous can inform us about retinal disease. Some of the pathways that we picked up were VEGF signaling, which you would expect, as well as superoxide degradation. I know Ilya Du at Wilmer has been working on that in terms of NRF2 and oxidative stress. And we also picked up Wnt2 beta-catenin signaling. My colleague Dave Antonetti and others have been working on that pathway as possible therapeutic for diabetic retinopathy. And then again, some axon guidance and notch signaling. So just recently, we did a much more thorough analysis of vitreous proteomics. This is a very complex data set. I'm not going to get into the details. I'm happy to ask questions about this later. So this involved 4 different 10-plexes. And what you can see on the right is a cluster analysis. And the samples are sorting by disease class. So blue over here is our control samples. And red, orange, and yellow are patients with proliferative diabetic retinopathy. And one of the caveats that we had to overcome is these are patients that have some blood in their vitreous. In fact, that's why they went to the operating room. And we were able to show that, in fact, the level of hemoglobin is quite low. and plasma proteins and red blood cell proteins are not skewing this clustering here. So the other big upshot of this manuscript was how many samples do you need to run in order to trust your pathway analysis? It's something that there's probably 20 to 23 papers about vitreous proteomics in the literature, and nobody had really stepped back and tried to figure out how many samples you would need to run to get biologically useful and reliable information. So it turns out, depending on the effect size that you're looking for, you probably need somewhere between 10 and 15 samples per group, which is fantastic. So by running the samples as a 10-plex, we obviously cut the cost by 10-fold. And so now we're set up to be able to do large-scale vitreous proteomics across several different retinal diseases. We actually have a very large vitreous biorepository, and we're sort of working through different disease states to identify novel pathways associated with various retinal diseases. So the next question our lab had is, why is the vitreous information rich? As I told you, this is typically surgical waste. It's not thought to have any function per se. And we went back to the original proteomics data set and looked at control vitreous and just did standard gene ontology analysis. And this is how we originally got interested in extracellular vesicles or exosomes. The gene ontology tree for the cellular component revealed that about 60 to 65% of the proteins in vitreous had been previously identified in exosomes or EVs by others. This was a totally new field to me at this time. It took us a while to sort this out and publish this manuscript. Before we could obviously publish a manuscript, bioinformatics analysis alone is not sufficient. So we went on a long quest to try to find and definitively identify the EVs in vitreous. Prior to our work, this is the type of analysis you would do with vitreous. Just a simple 2000 G pellet. Look on EM, you can see the collagen fibers and some cellular debris. If you spin the residual supernatant at 12,000 G and look at the pellet, again, you see collagen fibers and cellular debris. We were a little bit frustrated at that time. We admittedly didn't realize how buoyant EVs were. We finally looked at the supernatant and found that the supernatant was full of EVs based on TEM, as well as atomic force microscopy. So here you can see the original pellet, some cell debris and collagen fibers. And sitting in the supernatant, you can see a nice homogeneous population of EVs that are approximately 100 nanometers in diameter. So overall, we were able to publish that manuscript showing EVs are, the proteins in the vitreous are enriched for those that are associated with EVs. And when we do nanocyte on unfractionated human vitreous from healthy controls, we're able to show that there's like 1.3 billion exosomes per ml. There's about 4 to 4.5 mLs of vitreous in the human eye, so each eye has somewhere between 5 and 10 billion EVs. The cell source and function of these EVs is yet to be determined. We were one of the first groups to really comprehensively identify and quantify and determine the size distribution of these vesicles in human vitreous. We went back and looked at omen to see if there were any retinal disease causing proteins in human vitreous. And you can see here's a list of proteins that we found in our data set. The ones with an asterisk are EV associated proteins from exocarda. And I have an arrow here pointing to a protein called EFEMP1 or fibulin 3, which we find quite frequently in vitreous. It causes a macular dystrophy called Doyne honeycomb retinal dystrophy. And we'll be discussing that more later on in this presentation. So in conclusion for this part of the talk, vitreous contains critical molecular information. We believe the EVs are making the vitreous information rich, and therefore it's a plausible fluid for liquid biopsy of retinal disease. we've actually been successful isolating EVs from vitreous. Obviously, we want to do proteomics work on the EV fraction of vitreous. And we've actually made quite a bit of progress developing a vitreous biopsy device, a single-use disposable device to safely acquire vitreous in the clinical setting. So I'm going to change topics here and move on to what we do in my laboratory. And we focus on retinal pigment epithelial cells and their dysfunction in macular degeneration. So just to take a step back and introduce everybody to retinal pigment epithelial cells. When people think of the retina, they usually think of the inner retina and then obviously the photoreceptors. I put this picture up here on purpose to highlight our favorite cell, which are the retinal pigment epithelium. They are actively engaged and interact with the photoreceptor outer segments. They play critical roles in metabolism, recycling of photopigment, and so on. So these epithelial cells are terminally differentiated, extremely polarized, and in age-related macular degeneration, if you look at the literature, they become atrophic. They become smaller in size. They lose some of their pigment. More recently, in some clinical studies, migration of RPE cells into the retina has been shown to predict progression of dry AMD to wet AMD. And drusen, which I mentioned earlier, are one of the hallmark lesions of macular degeneration. You can see over here that you have the photoreceptors, retinal pigment epithelial cells, and they actually, these drusen are thought to be excrescences from these RPE cells. And sort of the central hypothesis of the lab is that we believe that these RPE cells are becoming less differentiated, moving along the continuum of the epithelial to mesenchymal transition process. As I mentioned, these cells are generally hyperpolarized, terminally differentiated, but they begin to acquire characteristics of mesenchymal cells where they migrate, proliferate, and secrete extracellular matrix proteins. In fact, other groups have done proteomics of human drusen and found that they're chock full of extracellular matrix proteins. And so our thought is that RPE cells may undergo EMT as a fundamental process underlying dry age-related macular degeneration. Just briefly, to support that hypothesis, we were able to acquire some post-mortem human retinal tissue and looked at the retinal pigment epithelial cells, and we were able to find ALEX, a well-known EV marker. And you can see that it co-localizes with fibulin. And this mound here is just a large soft drusen that's characteristic of dry age-related macular degeneration. And you can see that there are quite a bit of labeling of EV markers there. So the overall hypothesis of our group is that RPE cells transition from a well-differentiated state into a more mesenchymal-like phenotype. In doing so, they lose pigment, and some of the inciting factors to that would be aging, as in age-related macular degeneration, misfolded protein accumulation. One of the 34 known genetic risk factors is one of the more potent risk factors for AMD is HTRA1, thought to be involved in proteolysis of misfolded proteins. And these are obviously some of the key changes in EMT. So getting back to the fibulin protein I mentioned earlier, age-related macular degeneration is a very complex disease, 34 genetic loci. We've chosen to work with a simple model, which is a phenocopy of AMD. It's a autosomal dominant inherited macular dystrophy caused by a single point mutation in the fibulin 3 protein. Fibulin 3 is a secreted extracellular matrix protein. And it's thought to be, this mutation is thought to cause misfolding and inefficient secretion. These are some primary RPE cells. You can see here's wild type fibulin expression with a GFP tag. There's not much, there are punctate staining of fibulin within these cells. And as others have reported, the mutant version of fibulin gets, it's inefficiently secreted because of the misfolding phenomena induced by the point mutation. So we turn to the standard EMT assay, which is a scratch assay. And you can see by expressing wild type versus mutant fibulin 3, when you do the scratch, wild type cells are able to close the wound in a certain amount of time, around 18 minutes after induction of expression, whereas mutant cells are able to cross that wound much faster. More like, I'm sorry, the wild type cells take about 30 hours to close the wound, and the mutant cells take roughly 18 to 25 hours. They do start to proliferate, but that's not until approximately 36 hours in this assay. So we obviously wanted to get to the role of EVs in inducing EMT. To do that, we need to isolate EVs from these cells. For the proteomics experiments, we wanted a nice clean prep, so we turned to the iodixinol gradient. So here's the gradient here. You can see in lane 3, we have a nice enrichment of the buoyant EVs. This is our protocol here, which is pretty well known to everybody in the literature. You can see that in fraction 3, we find Alex, HSP70, flotilin, and luciferase tagged fibulin 3. To verify the quality of the prep, we used TEM. Here's wild type EV secreted from cells expressing wild type fibulin here, and here are the EVs secreted from cells expressing mutant fibulin. One of the things that sort of jumped out at us after looking at these images for a while is that there seems to be multiple distinct population of EVs, particularly in the cells expressing mutant fibulin. And so again, here's the EM of cells expressing mutant fibulin. Here's a cryo-EM. We're able to do a size distribution analysis, and in fact, there is a on average smaller size of EV secreted from mutant cells compared to wild type. The number of particles secreted, or the rate of secretion, is no different between wild type and mutant. And the protein per vesicle is also no different in EV secreted from cells expressing wild type or mutant fibulin 3. On the right, you can see a Western blot for Alex, CD81, and CD63. What's interesting to us is that when we express the mutant, the vesicles that are secreted look to have a reduction in the classic escort machinery that secretes classic 100 nanometer size exosomes. And on the right, you can see just some negative controls, again, just verifying our EV prep. So we did mass spec-based proteomics. We found about 2,800 proteins in each prep. They shared out 2,500 together, false discovery rate of 0.05. We used the mass spec to compare it to the Western blot to kind of validate the both systems in terms of Alex 01, flotilin, and Extin, fibulin 3, CD63, and CD81. And you can see that the protein abundances that we detect on Western blot are validated by mass spectrometry. I appreciate it's usually done in the reverse order, but that was just the way this study evolved. We did a typical pathway analysis using Advata, and we found that Hedgehog signaling was enriched in EVs from wild cells expressing wild type fibulin 3. So everything in blue is enriched in wild type. Anything in red is enriched in mutant fibulin 3. I know that a lot of folks find ribosomes in their EV preps, and we do as well. And we also found some lysosomal proteins, which I'm not quite sure what to make of that as of yet. Getting back to Hedgehog, that was interesting to us because Hedgehog is more abundant, more enriched in EV secreted by wild type fibulin 3 expressing cells, and Hedgehog is critically involved in the maintenance of RPE cells, retinal development, and it is neuroprotective effects on retina, promotes rod cone differentiation. In addition to Hedgehog, these were not significantly enriched, although they're pathways of interest. The ciliary tip plays a role in concert with Hedgehog to promote photoreceptor. It's an index of RPE cell terminal differentiation, and it works with Hedgehog to interact with the photoreceptors. EV assembly and escort proteins. So Alex is known here as PDCP6IP. These are more abundant in wild type EVs. And again, you can see the ribosomal proteins were found. Oddly, only one of them is found more abundantly in wild type proteins. I've searched the literature and I cannot find any regulatory protein for this ribosomal subunit. So unfortunately, at beta as well as IPA, they don't have a good pathway for EMT. Thankfully, this manuscript came out, I believe, in 2021, and they looked at 87 publications, high quality publications, and defined what they call the EMTome. We did a Venn diagram of the proteins from our EVs and found that we have an overlap of about 960 proteins, meaning that our EVs are enriched with proteins known to be involved in EMT. And even more impressive to us, one of these 87 manuscripts looked at the effect of TGF-beta induction of EMT, and 15 of the 22 genes in that manuscript were found to be altered in our EV groups. So that sort of triggered us to look at TGF-beta as a possible mediator of this process. And you can see here, when we look at the media from cells expressing wild type or mutant fibulin, there's more TGF-beta secreted by the cells expressing the mutant fibulin. When we remove the extracellular vesicles from the media, there still is a difference. There still is a little bit more free, non-EV bound TGF-beta in the media. Within the EVs themselves, from the EV isolation, that difference still exists. And critically, when we look at the proportion of free versus bound, we see that the majority of TGF-beta is actually bound to the EVs. And we have attempted to show that in our laboratory. I'm not sure. For those that are not aware, TGF-beta is a very complex protein, and the antibodies to look at that can be quite troublesome. But several other groups have shown that TGF-beta is actually on the surface of these EVs. So next we moved to, we went back to the scratch assay. This time the recipient cells are going to be just regular RPE cells. They're not expressing fibulin in any form. They're native, normal RPE cells. We're going to create our wound, and then we're going to add wild type EVs or mutant EVs, and then collect data for several days. So we wanted to see if the EVs were sufficient to induce EMT. And so what you see here is the same graph I showed you before, but again, these are regular RPEs being treated with two different populations of EVs. So wild type EVs, they do help to close the wound a little bit faster than the no EV treatment group, but the mutant EVs are sufficient to close the wound much quicker, and the half-life for wound closure is on the same scale as expressing the protein alone. You can see the calculation of the half-max over here. So we also looked at the recipient cells to see if we were getting any changes in RNA expression consistent with the induction of EMT. And treatment with TGF induces TGF-beta itself, but it also stimulates ZEB1, one of the key transcription factors involved in EMT that actually drives TGF-beta production. We didn't see any change in vimentin. I think that change might come a little bit later. This is just from a single time point. We also saw cadherin-2 change both in wild type and mutant treated cells. Moving on to the most critical experiment, we then took the same experimental protocol and we incubated the EVs with a pan TGF-beta blocking antibody prior to treatment. Again, TGF-beta is on the outside of these EVs, so The concept would be if you use a blocking antibody, you should be able to block the effect of EV-induced EMT. And so this is a graph on the left showing the wild-type vesicles. There's no impact because wild-type EVs don't really accelerate wound closure. But when we use mutant EVs, shown here in red and orange, you can see that the mutant EVs close the wound faster. When they're treated with a control IgG antibody, there's no effect. But when they're treated with the pan-TGF-beta blocking antibody, that's the darker maroon line here, it collapses back to the no EV treatment group. So this allows us to at least tentatively conclude that mutant EV induction of EMT is TGF-beta dependent. So in conclusion, RPE cells obviously secrete EVs. Expression of the mutant fibulin 3 alters the size of EVs. I think the escort system generally produces very homogeneous population of EVs. So I think in the context of activating the unfolded protein response, there's likely a different secretory system being used here. The cargo of the EVs that are being secreted, wild type versus mutant, is significantly different. EVs derived from the mutant cells are sufficient to induce EMT in recipient cells. And this induction, again, is dependent on TGF-beta. So just a couple of additional comments, and then we can open the floor to questions and have a conversation, open dialogue. So proteomic analysis of EVs. If anybody's interested in collaborating, we're more than happy to work with you. It's taken quite a bit of time and effort to get that going. We have a really robust system up and running now. We're looking for getting back to the role of vitreous EVs in retinal disease. One question our lab is struggling with is, where do all those EVs come from? Are they coming from plasma? Are they coming from various types of cells in the retina, ganglion cells, photoreceptors, RPE? So if anyone's aware of like a CRE-lock system with that has a GFP tagged Alex, that would be a really powerful tool for us to collaborate with. And as we mentioned at the beginning of the talk, about 2 years ago, we did start the Penn State Extracellular Vesicle Consortium. That's open to the public. It's the third Wednesday of every month. And if anyone's interested in joining the email distribution list, they can email me. And if anybody is interested or willing to speak, of course, we're always looking for folks to present. We have a mixed group of investigators, cancer, immunologists, neurodegenerative groups, as well as a couple of people working on retinal disease. Here's our acknowledgements. Obviously, can't do this with without all of our collaborators. So here's the core lab here. I have a couple of outstanding research associates, Yanjun Zhao and Stephanie Grillo. Jasmine Gethers is a MD PhD student. She will be completing her thesis soon. Sarah Weber has helped extensively with the vitreous proteomics. We're working on that device to acquire vitreous and fluid. Proteomics group that we're working with is from University of Michigan, as are the bioinformatics folks. They're outstanding. We're now working on converting, moving towards iPSC cells. We do have some really nice data showing iPS cells, differentiating them into terminally differentiated RPE, and we're starting to grow cells that have the Doyne honeycomb mutation paired with a cell line where they've used CRISPR to create a proper control. Some funding sources, and then as I mentioned before, a lot of collaborators helping us collect vitreous both at Penn State as well as Kellogg Eye Center at the University of Michigan. With that, we can open the floor to questions, and we can go from there.
Very good. Well, thank you so much, Jeff, for that presentation. I see that we have numerous people online. I see some people who have retina expertise, some people who are interested in the eye. And some questions are starting to come in the chat box here. I wanted to start though by going back to the first part of the presentation with the vitreous. Where are those EVs coming from, do you think? What cells are making them, and what other particles do you think might be present in that fluid that are maybe not EVs?
Yeah, that's a great question. So if you look at the size distribution on nanosite, you see EVs in the sort of the exosome size, 90 to 100 nanometers. You also see microvesicles, and then you also see some much larger EVs that are around the 500 nanometer size, more consistent with like an apoptotic bleb. Some of the proteins that I showed are thought to be uniquely secreted by RPE cells. People in the ophthalmology field are a little bit, how does a protein that's being secreted from the RPE make its way into the vitreous? But I think as this group would agree, I think almost every cell in the body secretes EVs, and we would really like to get a handle on exactly which cells are contributing to the EV population in the vitreous, particularly because just doing unbiased proteomics is so informative about retinal disease. So.
Very good. All right, well, let's go to our chat box then. And just to remind everyone, if you have a question, please just put it in the chat box so that I can call on you. And please be ready to turn on your microphone and your video if you'd like. So I've enabled all of that right now. So we can go first then to Padida Karimi, who has a question about the sizes of EVs.
Hello, can you hear me?
Yes, we can. Go ahead.
Okay. Thank you very much for your nice presentation. I have a question regarding microvesicles or medium-sized EVs. Do you also isolate, or did you isolate the microvesicles? I mean the RPE microvesicles, or just you focus on a small size EVs?
Yeah, we focus on the smaller EV group using filtration before we do ultracentrifugation. I think we're struggling with isolating subpopulations that are physically relevant, like most people in the field. Once we get the iPSC-derived RPE cells up and running that contain the fibula mutant versus a appropriate control, we really want to do proteomics to see if we can find a unique transmembrane protein that's expressed in cells and expressing the mutant fibulin. I think that's probably the easiest way to isolate the vesicles that are driving this differential effect in terms of EMT.
Thank you.
Thank you. Let's move on now to Alex Ryan.
Okay, thanks, Kenneth. Jeffrey, thanks very much for a great talk. I was wondering if you've either already looked at or have considered using tiers, instead of the vitreous fluid, right? For the vitreous fluid, the patient basically has to be in the operating room, but tiers can be collected more non-invasively. Have you looked at that? Have you considered looking at that?
So other groups have looked at tiers. I'm not sure if the EVs in tiers would be representative or have any relationship to the vesicles in vitreous fluid. The eye is basically a closed system, and the tiers are obviously on the outside. As I mentioned very briefly in my talk, I do agree with you that obtaining vitreous is invasive, generally only done in the operating room. We've actually been working for several years and have obtained significant funding, and we've been able to develop a single-use disposable, like a little handheld mini vitrectomy device that we believe will allow safe acquisition of vitreous in a clinical setting. But we're able to make that for very, I think the cost of goods now is around $25. And it has cutting action, just like the vitrectomy unit in the operating room. And so admittedly, the studies that we've done to date on proliferative diabetic retinopathy, we have additional manuscripts in preparation for retinal detachment and so on. Those are end stage or disease, and we'd really like to get into the patients that we're really interested in getting into, which make up the bulk of our population with vision losses, wet AMD and diabetic macular edema. Those patients do not have an indication to go to the operating room, but we would obviously love to be able to sample their vitreous in the clinical setting if we can do it in a safe manner. What's odd to me is I believe we're probably the only field in medicine using a very specific monoclonal antibody that costs around $2,000 per treatment just for the medicine, without actually measuring the target prior to treatment. Oncologists would never do this for the treatment of cancer. This would just never be done in any other field. We do it because we don't have a way to sample vitreous to see if the VEGF level is elevated. We honestly have no idea what threshold the VEGF would need to be elevated in order to have a response to treatment. So this is, admittedly, the field is sort of, it's moving in that direction, but we have a long way to go. So.
Okay. And just as a corollary, have you looked at the other fluid in the eye as well, the aqueous humor instead of the vitreous?
We have not looked at aqueous. Other groups have. There's a couple of conflicting reports out there. Some groups think that aqueous and vitreous are pretty similar ocular fluids. Other groups have shown that it's protein dependent. There's a correlation maybe for the majority of proteins, maybe 60–70%, but there are some proteins where there is no correlation in terms of their concentration, vitreous to aqueous. And then obviously it's going to depend on their lens status. If they've had cataract surgery, if they've had with a PC tear, or if it's a unicameral eye, then it's going to change the dynamics of the two different fluid spaces in the eye. So.
Thank you very much. I'll connect with you offline. Thank you.
Well, thanks for the questions, Alex. And I can't resist here just jumping in and sharing an anecdote, because I think that when my group was first starting to measure what we thought were EVs in a lot of different biofluids, we looked towards the eye and we got some tear fluid. It's very easy to just take a pipette tip, touch it there. You get a little bit, maybe even less than a microliter, but a very small amount. And what we found was that by using the the light scattering methods like NTA, we were getting a very high concentration of particles in that fluid, maybe higher than almost anything that we had looked at before. But what we found was that, of course, NTA is not specific to EVs. It's going to scatter from any particle that's there that's in the right size range. So we found that a lot of these particles were actually not EVs. They were lipid particles, but not extracellular vesicles, and that's because of the very high lipid concentrations there. So just we still don't know, and this is not a project that we pursued further, but we don't know what the percentage of those particles, what EVs represent as a percentage of those particles. So, all right. So we next have a question from Jim Honda. Jim, nice to see you. Please go ahead.
Nice to see you, Jeff. Nice to see you. Enjoyed your talk. I put one question, but I'm going to take the license to ask a couple. So with the RPE work that you've done, have you analyzed the EVs that are apical versus basally secreted? And part of the reason I'm asking that is your curiosity with all of these particles in the vitreous, which if it's from the RPE would be apically secreted, but you focused on drusenogenesis with the EVs going basally. And I'm wondering if you've analyzed any differences from apical versus basal secretion.
Yeah, I think that's a fantastic question. That's certainly an experiment that we've considered. For a couple of reasons, we haven't done it yet. Initially, working on with those transwells and just getting enough material to do proteomics on a small transwell was an issue. They now have much larger transwells that would make it more feasible from that perspective. And Michael Klingeborn, formerly at Duke, now out west, he has published at least one manuscript, maybe two, on differential proteomic, differential protein expression in apically versus basally secreted EVs. But I think that would be wonderful to do in a model of macular degeneration, like we're using with fibulin 3. I would guess that I would hypothesize that when the cells become less polarized, that there's going to be a major difference between the apical EVs in healthy, normal polarized cells. The apical basal secretion is going to be much different in polarized cells versus non-polarized cells. So I think part of the issue as these cells undergo EMT, at least in part, move down that spectrum to some degree, their directional secretion of these EVs is going to become disrupted.
Yeah, no, I agree. The other question I had for you is when I guess it was I think it was the fibulin 3 mutants. You had some overrepresented proteins that were related to lysosomes. Is that correct? So I'm curious. Do you think there's any connection? You know, with EVs, there's all these different forms, maybe some autophagy related particles that are released. Were there any proteins that gave you a clue in going in that direction?
Yeah, I went down that rabbit hole, and so if you obviously we're inducing the unfolded protein response, and we're inducing, there's 3 arms, and we're inducing the arm that is when activated can help induce EMT. What's interesting is I'm sorry, can you repeat the question one more time?
Were any of the overrepresented proteins related to autophagy?
So, you know, if you're an inefficiently folded protein, then, you know, I think I'm sort of guesstimating at numbers here, maybe 80 or 90% of that fibulin, you know, the chaperones in the ER are working really hard to fold it correctly, but maybe 80 or 90% of it needs to go towards the lysosome to get degraded. And so we started looking at autophagy, lysosome, and all of the various combinations of those terms. And, you know, some of those organelles are now, there's some sort of some secretory lysosomes or autophagolysosomes that can be secreted in various disease contexts. And yeah, I'm very curious, and I think you're right that some of these, like a subpopulation of these vesicles, might be related to the cell just trying to get rid of some of this extra misfolded protein that it just doesn't want to carry in its cell anymore, maybe through some sort of secreted autophagy type vesicle. So yeah, I agree. So.
Very good. Any follow-ups there, Jim?
No, I guess I agree with you, Jeff. I think if you extend that to AMD, I think maybe it's my simplistic view to drusen formation, but I sort of think of it as sort of a wastebasket of a bunch of junk. There's certainly the ApoB100 lipoproteins that contribute, and then there's a sort of inflammatory nidus debris. But I sort of think of it as waste products. So it may be other particles that are just trying to get rid of junk out of the RPE cell around which this inflammatory response develops, as opposed to more classic exosomes or EVs that are, that at least I like to think of it in terms of sort of cell-to-cell communication. So the context may change as the disease, as cells, for instance, go into EMT.
Yeah, I agree with that concept. I think this group in particular would think mostly of EVs as critical for cell-cell communication. I'm sure we all are aware that historically they were thought to be basically a way for cells to get rid of their waste and debris. And I think as the RPE cells go from self-respecting, fully differentiated, polarized cells, they might change their use of these secreted vesicles from cell-cell communication to under disequilibrium, trying to get rid of some cellular waste products that they might not normally need to get rid of.
Great. Yep. And just keep in mind that one cell's trash can be another cell's treasure too.
Exactly.
Yeah. So we have a question from Michael Pilatus. Mike, please go ahead.
Thanks, and thanks, Jeff, for an interesting talk. My question relates to the EVs and the vitreous again. Your work looked at EVs from the RPE being taken up by the RPE, and I wondered if you looked at or considered EV uptake in other cell types like Macular cells or photoreceptors.
Yeah, again, another outstanding question and concept, and I sort of touched on that a little bit. You know, I would love to you know, collaborate, or if I had more time and money. I honestly think that in a healthy eye, you know, the RPE cells are responsible for breaking down and digesting the outer segments, and you know, they have all these little microvilli that envelope these outer segments of the photoreceptors, and that's a lot of. First of all, that's a lot of lipid membrane to waste. I've kind of thought maybe the EVs are helping to recycle lipid bilayer back to the cell body of the photoreceptor, maybe, but maybe not just helping to recycle lipid bilayer because of energy considerations, but as I mentioned with the Hedgehog and ciliary tip pathway, those pathways have been shown to be involved in differentiation and maintenance of photoreceptor health. So we have not looked at that, but I think that's an incredibly interesting line of investigation for sure.
You know, my follow-up to that is they're not easy experiments to do. So.
No, yeah, so that's why we chose to focus on these other ones first. Exactly.
Very good. Well, it looks like we have come to the end of our questions in the chat box. And so I would like to thank you again, Jeff, for giving this presentation to us today. And I want to wish you all the best to you and your colleagues there at Penn State Hershey as you continue with your EV consortium. Thanks to everybody who joined today for all the good questions and comments. And we will look forward to seeing you at an upcoming EVClub. Take care, everyone.
Thanks so much, everybody. Just feel free to send me an email if you're interested in joining our group and or presenting. So we have a couple of holes in our schedule that we're looking to fill, and we'd be more than happy to have you speak, and that'd be wonderful. So I think collaborating and cross-pollinating is just fantastic for everybody. So thanks for having me, and have a great day.
Take care now.